conservation, and in general in California and especially in the Bay Area. My research is primarily in California native plants, but that extends to California native groups that have extended their distributions out of California, and especially in the Hawaiian Islands, which we don't tend to think of as having floristic affinities with California. And I'll want to talk a little bit about the history of that thought tonight. So I guess I will just go ahead and share my screen. So yeah, as mentioned, can you see that now?
Looks great. Thank you.
Oh, great. Yeah. So for 37 years now, I think I've been working on and off on one of these questions that's really fascinated me, and that ties in directly to California flora, and that really, I think, highlights the magnificent evolutionary potential of plants in general and Californian plants in particular. And all of this is against this backdrop of endangerment and destruction of habitat, et cetera, caused by anthropogenic factors. But I think there's still hopeful indications for at least some of the Hawaiian flora. If you think that the conservation challenges are significant in California, focus in on what's going on in Hawaii, and you'll feel a lot better about California. But there are some really heroic efforts going on in Hawaiian Islands to save some of these iconic plants that are relatives of some of our endemics here in California. And I like to think of these Hawaiian plants I'll be talking about tonight as honorary California endemics, even if some of my Hawaiian colleagues frown at that characterization. Okay. So when I talk about tarweeds, which is going to be a big focus tonight, tar plants is another name for this group. Traditionally, they've been called tarweeds, even though they are native plants. The name tarweed has been associated primarily with Californian members of the subtribe Madiinae, these true tarweeds like Railardella, Holocarpa, Calicodenia. A lot of our really spectacular summer flowering flora in California is part of this group. But about 20 years ago, we published a paper showing that actually the California tarweeds are part of a larger, primarily Californian radiation that includes the woolly sunflowers, groups like goldfields and the true woolly sunflowers and Eriophyllum, as well as some other genera, a few shown here. And Arnica, the medicinal genus Arnica, is actually sister to the true tarweeds. This is a circumboreal group, but the diploids are primarily in California, or at least California Floristic Province. And then some things like canyon sunflower, Venegasea, and Hulsea found at higher elevations. These make up a broader tribe called the tribe Madiinae, the tarweed tribe. But traditionally and still today, the tarweed subtribe within the sunflower family, or Asteraceae or Compositae, is this group of about more than 90 species in California, about 21 genera in California, that we've resolved represent 4 major lineages that are almost endemic to the California Floristic Province, and most of the species are, in fact, the vast majority. And these represent the Leia lineage, Leia and Legophylla, the Hemizonia lineage with Hemizonia and Blepharozonia, Calicodenia lineage with Dianandra, as well as Osmodinia and Centromedia, for example. And then the Madiinae lineage, which we're going to focus on a lot tonight, that includes a number of genera. And this is just with regard to the Californian diversity. I'll get to the Hawaiian diversity in a moment. But this is one of our really iconic Californian groups. And we focused on all of these genera. I just want to briefly mention Leia is not part of our summer flora. It's part of our spring flora. And we've done quite a lot of work in this group, and we continue to, for example, in the Leia glandulosa complex. This is a little kaleidoscope of diversity of lineages just within Leia glandulosa and this descendant lineage called Leia discoidea, well named because it lacks ray flowers and is a serpentine endemic in the south inner coast ranges that represents an example of accelerated divergence in these unusual serpentine soils that, of course, are a source of a lot of our endemic diversity in California. And just this year, we've recognized this new species, Leia erubescens, from the back dunes of San Luis Obispo and Santa Barbara counties, where it was hiding in plain sight, had been previously collected, but clearly an outlier to this group, and named erubescens for these blushing ray flowers. Erubescens referring to blushing or reddening. In addition to Leia, these other major lineages, these three lineages here, are predominantly summer flowering tarweeds, although there are spring flowering populations and species in these groups. And they represented a major part of our flora. I'm just going to focus in right now, though, on the Madiinae group. Madiinae elegans shown here, which is exemplary of our summer flora, as well as having spring flowering populations. And these are really beautiful garden plants. They really are bee magnets, some of the best magnets for bringing in native bees into gardens, Madiinae elegans. And it's very showy, and it's self-incompatible. It's an obligate outcrosser that must outcross in order to reproduce. Whereas much of Madiinae includes plants that are self-compatible, that are sulfurs, like Madiinae exigua, which is at the extreme of lack of showiness, with corollas that are almost invisible here, and which reproduce by selfing to a large extent, are more ruderal in their ecology. Also in this Madiinae lineage, we have some segregate genera, like Harmonia, which is a group in the North Coast ranges, pretty close to Marin County. Harmonia nutans in the volcanic ash exposures in Napa and Sonoma County. And that is in turn sister to all these serpentine endemics that make up the rest of the genus that occur across serpentine islands in the North Coast ranges, and seem to represent one of our best examples in the California flora of island hopping and diversification in regard to moving into different serpentine exposures that are geographically isolated, never been connected. And we have these beautiful endemics, all of which are 1B species within California Native Plant Society's rare plant inventory, quite rare. But these aren't the only examples of insular evolution in the subtribe Madiinae, or the tarweeds. In addition, I mentioned in Leia, we have these edaphic endemics and these islands of unusual soils. We actually have real island endemics in this group, and some of them are in the California islands in the genus Deonandra, which unfortunately tonight I won't have time to talk about, but others are in the Hawaiian Islands, and they are quite spectacular. If you haven't seen them, I do recommend a trip to the Hawaiian Islands, especially to Haleakala National Park, where you can see the Haleakala silversword seen here on the left, which gets well over 1 meter tall. I've seen some individuals approaching 2 meters. And I'll talk more about them in a moment, but this idea that there was a relationship between these Hawaiian endemic silver swords, the true silver swords being in this genus Argyrosiphium, literally silver sword, was first proposed by the great Harvard botanist Asa Gray in 1852. This is before Darwin's theory of evolution was published. He was quite a champion of Darwin's theory in America. And he placed the genus Argyrozyphium in the California tarweed subtribe Madiinae, along with plants like the Leas and the Madias. This idea gained a lot of credibility because he was the most influential taxonomic botanist in America at the time. But it was difficult to reconcile with the notions of biogeography at the time. And actually, Darwin's co-author of the theory of evolution by natural selection, Alfred Russel Wallace, who is also often considered the founder of the field of island biogeography, struggled with this relationship in his famous book, Island Life, published in its first edition in 1880, where he said, "We may look upon the compositae, the sunflower family, as representing the most ancient portion of the existing flora of the Hawaiian Islands, carrying us back to a very remote period when the facilities for communication with America were greater than they are now." So he figured there had to have been some mechanism to allow plants from California to get to the Hawaiian Islands, and suggested there were early bathymetric data, very sketchy, suggesting that there might have been submerged islands that were once above sea level between Hawaii and California. And also thought that this relationship had to be extremely ancient because these silver swords were so different from any other composite that and given the gradual nature of the evolutionary process as they understood it at the time, it must have taken a phenomenal amount of time for those things to evolve. The idea, though, that there was some islands in between Hawaii and North America that would allow for island hopping of short distances to get there was wiped away by better understanding of seafloor topography. And here you can see the Pacific Basin floor, and notice that there are no islands that can be hypothesized to have ever been above sea level, except very close to California and the California Islands, of course. And this is a distance of about 3,500 kilometers, about 2,000 miles. And so botanists throughout the early half of the 20th century really struggled with the possibility of any kind of a floristic connection between North America and Hawaii. To the southeast or to the southwest in Australasia or Southeast Asia, there was, of course, a number of island groups, and a sense by some botanists, at least, there might even have been a continental platform that existed at one time and that was now sunken, and that there might have been an overland route to Hawaii. But no one could come up with a way to get plants from North America to Hawaii. And it was a real conundrum because long distance dispersal of that kind of intercontinental or oceanic magnitude was not considered feasible by most botanists at the time. And even the great botanist David Keck, who is famous in California botany for his contributions to our understanding of many plant groups, including the California tarweeds, which he studied extensively with Clausen and Heise as part of the famous Clausen, Keck, and Heise team out of Stanford, suggested in his monograph of the Hawaiian silver swords in 1936 that the affinities of these genera of the silver sword alliance are to be found to the south or southwest in the Pacific. That is, he suggested, for example, Robinsonia, which is a genus of the Senecio tribe that's endemic to the Juan Fernandez Islands off the coast of Chile, or Bedfordia, which is endemic to Australia, as among the closest relatives of the Hawaiian silver swords, and basically concluded that by thus divorcing the silver swords and Argyrozyphium from the American tarweed genera to which it has been thought related, the most persistently proposed connection between the ancient element in the Hawaiian flora and the New World has been shattered. And so now I want to segue away from this early thinking to give you a context for the geology of Hawaii, to put what we know now into better context. So the Hawaiian Islands, as you may well be aware, is a hotspot archipelago, which means that there's a more or less stationary hotspot at about this position off the southeast coast of the big island of Hawaii that has been generating islands for about 29 million, well, for longer than 30 million years, but for about 29 million years, the direction of movement of the Pacific plate over that hotspot has been northwesterly in this direction, such that we have a linear chain of islands of increasing age as we go from Hawaii at about 1 million years to the Maui Nui complex, which includes Molokai and Lanai at about 2 to 3 million years, Oahu, where Honolulu is located, about 4 million years, and Kauai, the oldest high island, at about 6 million years. But if we look out to the west, there's a long chain of sunken seamounts, atolls, and islets, ending with Kure and Midway, Kure being at about 29 million years ago, that are also in this progression but are quite small today. If an organism arrived as long ago as 29 million years ago, though, in this chain, if it was capable of dispersing to the east to new islands as they emerged, it could potentially have persisted for that long a period of time. So there is some credibility to the possibility that a Hawaiian organismal group, a plant group, for example, could be as old as 29 million years if it was able to do that. So just bear that in mind as we go forward. But these are the high islands of the Hawaiian chain that exist today. And this Hawaiian Silversword Alliance, as we call it, is a group of 36, or I guess 36 or so species, about 45 taxa, I should say, including subspecies, that are comprised of are found in 3 different genera: Argyrosiphium, the true silver swords and green swords, which I already introduced, that have these fleshy basal leaves and produce these large capitulescences that can have up to 600 capitula or flowering heads. In the case of the Haleakala silver sword, these are all found at higher elevations on the Big Island of Hawaii and on Maui, both East and West Maui. And then at the other end of the chain of the high islands is the Hawaiian genus Wilksia, which is comprised of only 2 species. These are also rosette plants, but they are fibrous rather than fleshy-leaved rosettes. They have anatomy very similar to that of a yucca. And these rosettes are borne on pole-like stems that can reach up to about 15 feet tall or so, in Wilksia genosiphium shown here. And they're found in drier slopes of Kauai. And then finally, the genus Dubaudia, which is the most diverse group that has the vast majority of the remaining taxa in the group. And these include trees, like you can see here, Dubaudia knudsenii from wet forests of Kauai. A liana or woody vine, Dubaudia latifolia from mesic forests of Kauai. Dubaudia scabra, which is a mat-forming pioneer colonist of young lava flows on the younger islands. And Dubaudia waialeale, which is a cushion plant found on the summit bog of Waialeale here on Kauai, which is one of the wettest places on Earth. So we have, and then there are a wide array of different forms of shrubs and other trees in the group. So we have a lot of habit diversity in terms of life form. We have a lot of diversity of ecological settings, from some of the driest habitats of the islands, some of the wettest habitats on Earth. So a major example of adaptive radiation, this phenomenon where evolution is thought to be relatively unconstrained due to a lot of ecological opportunities and a high ecological propensity of the organisms involved. So this group of plants was studied extensively in the second half of the 20th century by Sherwin Carlquist and Don Cayos and students. So Sherwin Carlquist was at the Rancho Santa Ana Botanic Garden, now the California Botanic Garden, for his career, most of his career. And he was a plant anatomist who unfortunately just passed away last December, and did extensive anatomical work in the Silversword Alliance and the California tarweeds, concluding that in fact Asa Gray was correct, that the silverswords and relatives were in fact closely related to California tarweeds. And Don Cayos was an experimentalist that did a lot of crossing studies between members of the Silversword Alliance, and with one of his first graduate students, Jerry Carr, who's still living. Don unfortunately died in April. He was my major professor as well, but relatively late in his career. Jerry Carr, who's still alive, did a lot of work with Don in looking at the cytogenetics, the chromosomal genetics of this group, and laid an important foundation of understanding of the group's genetics at that time. So one of Sherwin Carlquist's major contributions in island biology was basically to crystallize our understanding of the importance of birds in moving plants to islands. And from his famous book, Island Biology, published in 1965, which is his first major book on islands of several, Sherwin concluded that dispersal of fruits and seeds by migratory birds is probably the method by which most groups of flowering plants arrived on the high islands of the Pacific, which are some of the most isolated islands in the world. And this is actually a figure from Ballard and Seitzma's paper on Hawaiian violets, where they nicely summarize the proportion of species of Hawaiian of birds that regularly migrate to the Hawaiian Islands from different parts of the Pacific Rim. And you can see that North America has a high proportion of birds that travel to the Hawaiian Islands on a regular basis. And the Pacific golden plover, for example, is a famous example of this type of migratory route from the more northerly part of the continent. And so Sherwin Carlquist suggested that that groups of Hawaiian plants could easily have been transported by birds to the islands, even though that's an amazing distance. If we look at the flora of the Hawaiian Islands, we find some major indications of floristic isolation there that are consistent with the idea of an extensive amount of evolutionary change in the islands. So greater than 90% of Hawaiian flowering plants are endemic to the Hawaiian archipelago or found nowhere else in the world. So there are about 1,000 native plant species in Hawaii, and about 90% of them occur nowhere else in the world naturally. And when we look at the composition of that flora, it's highly disharmonic. And by that we mean that the representation of plant groups in the Hawaiian flora is not very, doesn't seem to represent a random sample of the groups that are found in the source areas around the Pacific Rim. It's highly biased toward groups that are more readily dispersible. For example, gymnosperms, including conifers, are absent altogether in the native flora. And the real kicker here in terms of evolutionary importance is, and this is something we now only know on the basis of a lot of recent phylogenetic work looking at the relationships of Hawaiian plants, of the roughly 250 or so founders that we have to hypothesize were natural founders for the native flora, only around 10% of those founders account for about 50% of the species of flowering plants. So this means that a minority of the founders in the Hawaiian flora gave rise to half of the native flowering plant diversity. So there's been a disproportionate amount of evolutionary activity on the part of some plant groups since they arrived in the Hawaiian Islands. And this could have a number of explanations. For one, it might represent groups that got there early when there was a lot of open ecological space, when there was a lot of ecological opportunity. It could also just reflect a greater evolutionary propensity of particular plant groups, or both of those things in addition to other factors. So getting back to the silver swords and the California tarweeds, when we look at the relationships of the Hawaiian silver swords, the results from molecular phylogenetic work that we did many years ago basically corroborates the hypothesis of Asa Gray and the idea of Sherwin Carlquist that the silver sword alliance is most closely related to California tarweeds. But not only that, the silver sword alliance is not the sister group of all the California tarweeds. It's not as if they shared an ancient common ancestor and then underwent diversification separately at a later time. The silver swords are actually nested within the California tarweed radiation. I mean, if we want to get really strict about it, the silver swords are California tarweeds in the sense that the California tarweeds had already diversified extensively in California or Western North America into those major lineages I mentioned that are shown here in different colors, like the Hemizonia lineage, the Madiinae lineage, the Leia lineage, Californian lineage. But the silver swords are nested within one of those lineages, the Madiinae lineage. So they are part of that group. And you can see here representatives of Argyroxiphium, Wilksia, and Deboudia forming a natural group nested within this Madiinae lineage. The root of this tree is down here and the tips are out here on the right. So it's a tree lying on its side. What this tells us is that pretty ironclad evidence that the origin of the Silver Sword Alliance was from Western North America and would have represented a dispersal event there. And also that woodiness in this Hawaiian group, which is pervasive, is derived from herbaceousness, which we find throughout the Madiinae lineage and in most of the lineages of the California tarweeds. So woodiness being secondarily derived from herbaceousness, that used to be anatomical heresy to suggest such a thing. But Sherwin Carlquist championed that idea as well, which was actually first proposed by Charles Darwin and also Alfred Russel Wallace that I mentioned earlier, the founder of island biogeography. But it fell out of favor with plant anatomists. But this example, as well as others that came later from studies of island groups, really reinforced Carlquist's idea that, in fact, woodiness can be derived from herbaceousness. And if we look at the closest relatives of Hawaiian silver swords in California, they're intriguing plants because they show some features that are quite suggestive of what the ancestor might have looked like. A lot of the lineages branching off prior to the silver sword origin are perennial. They're not woody, but they are rhizomatous. Things like Chiosea, which is a monotypic genus that occurs in wet meadows and boggy meadows in the Sierra Nevada North Coast and Klamath ranges, higher elevations. Anisocarpus, which includes the woodland media of the Bay Area and other parts of California, occurring in understories of forested or woodland areas. And Anisocarpus scabritus here, which is a subalpine plant of exposed metamorphic scree. And there are others as well in this, like Carlquisti, I'll show you in a moment, which bring home the possibility that there was a lot of evolutionary lability already in this Californian group of relatives that occupy a wide array of different kinds of habitats. The Madiinae lineage, of course, also occurring in really dry habitats, but we have everything from subalpine dry situations and exposed situations to forests and meadows, a lot of ecological niches filled in this group. And the perenniality, this rhizomatousness, would have allowed the ancestor to become established vegetatively before becoming reproductively active with regard to a lot of cross-fertilization, because one of the intriguing things about the Silver Sword Alliance is that they're self-incompatible. They are obligate outcrossers, and they appear to be primitively so, a real exception to Baker's rule, which dictates that long-distance colonists will tend to be self-compatible for the obvious reason that if only one gets there, they won't be able to establish a new population. But in the case of the Silver Sword Alliance, either multiple individuals arrived at the same time at the same place, or else we probably had a perennial established that could vegetatively reproduce until there were enough mutations for new self-incompatibility alleles to develop to allow them to resume sexual reproduction. So it's still one of the great mysteries of this group, but there are some hints here as to what may have occurred. As far as getting there, these plants are well equipped for bird dispersal. If you've ever walked through a field of tarweeds in the summertime, for example, a field of Madiinae elegans, like you see here when at the shattering stage in fruit, you'll understand that the ray fruits, the blackened ray fruits here, are surrounded by sticky involucral bracts or phyllaries that tightly adhere to material like socks or pant legs. They also can attach to fur or feathers as well. And in the perennials within the Madiinae lineage, the disc flowers have pistillate function as well. And they also have a pappus, these modified sepals that are bristle-like and that will allow the fruit to lock in at about this position if it penetrates from the tip. They can also lock in along the fruit wall here because of these upwardly pointing hairs. This is a disc fruit of Carlquistia, which is a perennial relative of the Silver Sword Alliance, and Chioscia that I just showed you from the wet meadows here. And this is their F1 hybrid, which is sterile, but has sterile fruits here. So the real clincher with regard to this relationship early on was that it was possible to make a hybrid between these perennial members of the Madiinae lineage and members of the Silver Sword Alliance. So during my dissertation work, once these molecular data, these DNA data became available to indicate that, yes, the Silver Sword Alliance is a natural group and it's closely related to members of this Madiinae lineage, I attempted to make a cross between Carlquistia murei, which at that time was called Raylardella murei, and this is a mat-forming perennial herb, a rhizomatous herb from high montane situations in the southern Sierra Nevada and the Santa Lucia range of Monterey County, and Devatia levigata, which we had growing in the greenhouse Davis at the time, which is a mesic to wet forest, large shrub to small tree. And the cross was incredibly successful. There were many, many full fruits. And this is one of the very vigorous but very confused hybrids that resulted from that grew up to the top of the greenhouse and back down again. Didn't really know what kind of life form it wanted to be, but these were vigorous plants that persisted. We grew them for quite a while, but they were quite sterile as well. They showed hybrid vigor, but also like a mule, they were quite sterile. And we made a number of other crosses with other perennial members of this Madiinae lineage that were also successful with other members of the Silver Sword Alliance. Subsequently, a number of other studies were undertaken that caused us to understand that there are a number of other groups in the Hawaiian flora that have a West Coast ancestry here in North America. And the first of these that was studied were the Hawaiian sanicles, section sandwichenses in the genus Sanicula and Apiaceae or Umbelliferae. And there aren't nearly as many species of these as there are members of the Silver Sword Alliance, but they're quite diverse ecologically and quite interesting. And a postdoc, my first postdoc at Berkeley, Pablo Vargas, now at the Royal Botanic Garden in Madrid, for his postdoc work, looked at this relationship, and this was in collaboration with our dearly departed colleague, Lincoln Constance, the Apiaceae expert at Berkeley. And what we found was that not only are the Hawaiian sanicles closely related to the California santicles, but like the silver swords, are nested within the California santicles, most closely related to a small group of species of Californian species, namely Senicula arguta, laciniata, and the footsteps of spring, Senicula arctopoides, that you could consider the footsteps to Hawaii. Also, the Hawaiian mints, which are the second most diverse lineage of Hawaiian angiosperms, with about 60 species in 3 genera, like the Hawaiian species of the Silver Sword Alliance, namely Philostygia, Haplostachys, and Stenogony. These 3 genera turn out to be, on the basis of work by Lindquist and Albert, nested among the West Coast Stachys. So plants like Stachys chamasonis and our other natives in the California flora are very close relatives of the Hawaiian mints, which are a magnificent group. The Hawaiian violets, which are incredible plants that include these little treelets, miniature tree-like forms that were thought to be closely related to woody violets elsewhere in the tropical regions of the world, turn out to be closely related to West Coast West Coast violets as well, based on work by Ballard and Seitzma, and some subsequent work by other teams as well, including these gentlemen. And the Hawaiian Caryophyllaceae, the Hawaiian Shaidias, including Alcinodendron, very diverse group, turn out to be of American origin too, but not from members of the Californian flora. So there's a big American and a considerable Californian influence in the Hawaiian flora, despite the earlier thinking that California contributed very little. But when you look at the climatic situations in these high volcanoes, especially in the highest peaks on Maui and the Big Island, there's a considerable overlap of the climate with part of the climate of the California Floristic province, particularly in the northwestern part in the Klamath Ranges. So we do have a lot of climatic similarity today, and presumably we had climatic similarity in the past as well, that allowed for this temperate origin of technically tropical but ecologically temperate plants in these higher elevation habitats, even though some of these groups have moved downslope as they've evolved new adaptations. So I just want to summarize some of our basic evolutionary findings for the Silver Sword Alliance for you, just these major bullet points. That what we found in studying these plants over the years is that diversification or speciation has mostly happened within individual islands of the Hawaiian chain, with relatively few dispersal events between islands. And these diversification events have been marked by major habitat shifts and sometimes major changes in plant life form as well. And one of the most extreme examples involves some of the plants I've already shown you. And this is on Kauai, where there's been a miniature radiation involving members of the genera Wilkesii and Dabaudia. So I mentioned Wilkesii, which these 2 species of xerophytes that grow on the west slope of Kauai, and it look like Dr. Seuss's truffula trees with these strange discoid, long peduncled heads on these rosettes and long stalks here, easily seen on the Iliao Loop Trail of Waimea Canyon, if you ever get to the Grand Canyon of Kauai. And as I mentioned, these are fibrous leaved rosette plants with perfectly parallel leaf venation. And it turns out they're not most closely related to the other rosette plants in Argyrosiphium, the true silver swords and green swords, which had been some earlier thinking, but instead are most closely related to a subset of the species of Dabaudia. And these are all endemics to Kauai as well, including Dabaudia railardioides, which is a scandent shrub, this clambering shrub that grows through the understory of wet forests and emerges in openings. Also, Dabaudia paleata is a close relative. This is a more compact shrub of open exposed bog situations, these acidic bogs that pose different challenges to plant growth. And also Debaudia latifolia, the only liana or woody vine in the group, which has highly reticulate net-like venation of its leaves, as different from the leaves of Wilksia as a typical eudicot is from a monocot. This also appears to be part of this same lineage and is has a different habit growing in mesic forests, yet a different type of forest on Kauai. So this is an example of a major radiation within the oldest modern high island. And what we've recently found in this paper that came out last October is that a number of recently described species on Kauai turn out to be very closely related to one another. So we have this newly resolved endemic Kauai lineage. The Debaudia lyalli I mentioned, the cushion plant from the summit bog of Kauai, and latifolia or levigata, the species that was first successfully hybridized to a California tarweed. These have been described long ago, but these other four species have been described since the 1980s. And in part due to the very intrepid and dedicated field work of Kauaian botanists like Ken Wood, shown here, for whom Debaudia kenwoodii was named, and unfortunately now appears to be extinct. It grew on cliff faces around the Kalalau Valley, where Hurricane Iniki in the early 90s really stripped the vegetation off of those cliff faces with average wind speeds of over 180 miles per hour, really devastating hurricane. And those habitats that were basically protected from the feral goats, pigs, etc., deer, that devastate much of the vegetation in these wetter parts of Kauai and where they were protected. So unfortunately, those habitats that couldn't be reached by those ungulates, and they were then subjected to recolonization by weedy species. So this is a sad story, but the members of the National Tropical Botanical Garden have done a lot of work to try to save the Kauaian flora, and they deserve a lot of credit for that effort. Also, I want to point out the diversification or speciation of the Silver Sword Alliance has occurred at an exceptionally high rate, and it's happened within the time frame of the modern high islands. As I mentioned, the Hawaiian Islands extend out to Kure and Midway, which are at about 29 million years maximum age. And anything that got dispersed out there that long ago could potentially hang around and not be submerged if it was able to disperse to younger islands as they emerged. But that doesn't appear to be the case for the Silver Sword Alliance. And Mike Sanderson and I published a paper about 25 years ago now where we were able to publish a calibrated phylogeny for the Silver Sword Alliance shown here and close perennial relatives in California, the Madiinae lineage here. And what we found was we used a maximum calibration. So the maximum age of the Silver Sword Alliance most recent common ancestor is about the age of Kauai, the oldest high island. And so this suggested that the group was younger than might have been expected, and certainly younger than Wallace and others like Kex thought it was. And recently in 2018, we published with Michael Landis and Will Freeman, Michael leading up this study, we published a new phylogenetic analysis of the Silver Sword Alliance, including close relatives in California. They gave us a more refined estimate of the age of the Silver Sword Alliance at only about 3.5 million years ago. And the nice thing here is we were able to actually estimate the age of the colonization event in Hawaii, which was dated subsequent to the emergence of Kauai. So this nailed down more clearly that this group is of contemporary Hawaiian origin and doesn't date back to the Leeward Islands. And the group then might be expected to follow the progression rule of Hawaiian biogeography. And this is a simple historical biogeographic hypothesis for lineages that diversified across the archipelago and that arrived early. And Carlquist, this was a hypothesis from 1995 by Vicky Funk and Warren Wagner, major contributors to the Hawaiian understanding of the Hawaiian flora. But Sherwin Carlquist at an earlier time pointed out there seems little doubt that Kauai has been a major source area for the rest of the archipelago. And the idea of Funk and Wagner was that organisms that arrived early on Kauai would have dispersed down the chain from older to younger islands. And we should see a pattern of relationships that looks something like this, where this is the root of the tree. These are tips of the tree, with Hawaiian groups being highly nested within this overall phylogeny. And that's exactly what we see in the Silver Sword Alliance. Interestingly, in Argyroxiphium, the true silver swords and green swords endemic to Maui and the Big Island, we actually have evidence from the length of the branch of this Argyroxiphium lineage that they date back to Kauai, where there is no longer any high elevation habitat to the extent that there is on Maui and the Big Island today. These youngest islands, it's extensively eroded and subsided, but presumably they did occur on these older islands at one time, or at least on Kauai. There's another lineage that occurs not on Kauai, but on these younger islands. And again, the date of this lineage is consistent with a Kauai origin, and it appears to have dispersed predominantly from older to younger islands, although our resolution here, whether it went to Maui first and then back to Oahu, or Oahu and then on to Maui, it's a little unclear. But we have one example here that's very ironclad of dispersal from the Big Island back to Maui, an older island. And this involves the species Dabaudia linearis. This is the endemic Hawaiian subspecies of that group, which appears to have formed first. And then based on comparing chloroplast DNA relationships, there is a genome in the chloroplast organelle, as well as the genome in the nuclear, in the nucleus. We compared the chloroplast tree that we reconstructed with the nuclear tree, and there was conflict that suggested that what had happened is that this species of this Dabaudia linearis had dispersed back to Maui, and once it arrived there, it hybridized with the resident species, but remained distinct, even though it's somewhat different morphologically, treated as a different subspecies, which might represent influence from hybridization, and captured the chloroplast of the resident species there. So back dispersal can occur. And in the two most widespread species of the Silver Sword Alliance, Dabaudia laxa and Dabaudia plantaginea, shown here, we have evidence that is consistent with two dispersals out of Kauai for these two species, colonizing different parts of the archipelago. So that kind of brings to light that there is more dispersal than we thought, but still predominantly dispersal within our diversification within islands. There also is evidence for a hybrid origin of the Silver Sword Alliance involving two still living lineages of California tarweeds. And this evidence comes from a study published in 1999 that involved three labs, my lab, Rob Robichaux's lab, and Michael Peruginen's lab, where we found what we knew already was that in all of the Hawaiian species, there are two separate genomes. So they're tetraploid. They're not diploid like us. They have two distinct genomes. And those just different genomes, when we cloned out this, when the copies of paralogous genes that are the result of, that are from those different genomes, but comparable genes, were pulled out and sequenced and included in phylogenetic analyses, what was found, treating those different genomes as different taxa in the analysis, what was found is that one of the Hawaiian genomes found across all the Silver Sword Alliance is most closely related to one of the lineages of the Madiinae group in an Isocarpus, and the other lineage, or the other genome in the Hawaiian Silver Sword Alliance that's found throughout the group is mostly closely related to a different lineage in the Madiinae lineage. So the simplest hypothesis, in our opinion, is that there was an allopolyploid or hybrid polyploid, tetraploid formed in California between those living lineages of members of the Madiinae group, and that hybrid polyploid was transported to Hawaii by a bird, became established there, underwent diversification, and at some point, those hybrid polyploids became extinct in California. The alternative hypothesis is that both Californian lineages made it to Hawaii, hybridized there, and formed this hybrid polyploid, and then the diploids went extinct in Hawaii. Both hypotheses could be regarded as having exceedingly low probability. And so, you could argue it one way or the other, but in any case, it appears that the Silver Sword Alliance has a hybrid origin involving different living lineages of the California Madiinae group. And it's still possible today to make that hybrid after these results were, well, even before those results became evident. I had been making a lot of crossing attempts in the California Madiinae lineage, and a cross that I made between an Isocarpus scabridus and Carlquistia murray that represent those two different genomes in the Hawaiian silver swords was successful. And here you can see the first generation hybrid, which is quite viable, but the only viable pollen in that individual, and it's a small fraction of the pollen that's viable, is diploid, not haploid, because it has 4 pores rather than 3. They're quite large, and in composite, tetraporate grains of pollen are typically diploid in constitution. So these pollen grains would be the raw material for forming a tetraploid if they got together with a tetraploid egg, which presumably also is being formed here from a failure in meiotic divisions during development of sporogenesis here. And so we do have evidence that even millions of years after the Silver Sword Alliance ancestor formed, this hybrid is still possible to form, and it gives us biological evidence for the viability of this idea. So I'm running out of time here. In fact, I've run out of time, but I do want to mention a couple of things. There is a lot of potential for hybridization within the Silver Sword Alliance. And I just want to mention that this group, 43 different hybrid combinations have been documented in nature within the Silver Sword Alliance, more all the time. And one of the most extreme is the hybrid between the Haleakala silver sword shown here in East Maui and the Haleakala kupa-oa, Debaudium menziesii, which is a very common plant at high elevations where we find the silver sword. These represent different lineages in the Silver Sword Alliance that are relatively distantly related, but they're part of the same radiation. And if you hike down the Sliding Sands Trail in Haleakala, it's easy to find their F1 hybrid growing there, as you see here, and which is very easily recognizable as something different. It looks very intermediate between them. And Jerry Carr actually back-crossed an F1 produced in the greenhouse to this kupa-oa parent, and within 2 generations produced a plant that was fully fertile. So even though the hybrid has reduced fertility due to chromosomal rearrangements that I don't have time to get into, there's still potential for gene flow between even the least interfertile species in the Silver Sword Alliance. And this is just a diagram showing all the different genomic arrangements present in the Silver Sword Alliance. And these lines indicate hybrids formed in all these different combinations. You can cross the trees with the shrubs, with the vine, with the rosette plant, any combination. The hybridization is readily achieved. The hybrids have some level of fertility. And when we look at the relationships within the Silver Sword Alliance, this is a busy diagram, but what I want to focus your attention on is just these blocks here. This is a nuclear phylogeny facing to the right and a chloroplast phylogeny facing to the left, involving the very same DNA accessions from all the species in the Silver Sword Alliance. What we see here is the Deboutia laxa, represented by all of these accessions here, is a natural group according to nuclear data, but it's scattered all over the tree based on chloroplast data. And these are branches separated all the way to the base of the tree. That's suggestive of chloroplast capture by hybridization that's resulted in a genomic change in the constitution of these plants as a result of hybridization. Deboutia plantaginea, similarly, a natural group based on nuclear DNA. Based on chloroplast DNA, they're scattered across the tree. Again, presumably and likely due to hybridization. And Deboutia knudsenii, again, a natural group based on nuclear DNA. Chloroplast DNA suggests they come out in different major clades or lineages of the chloroplast phylogeny, probably due to hybridization. So hybridization has probably played a role in the group. We're trying to get a better handle on that now, looking at genes across the entire genome, nuclear genome. Finally, I just want to say that evolution of most leaf functional traits, when they're looked at in terms of how the plants bearing those different leaf functional traits are related based on phylogenetic data, there appears to be very little constraint. That is, it appears that the adaptive radiation has been pretty much unhindered by any kind of evolutionary constraints that might result in some inability to move into different trait space or evolve different kinds of features. This is work by Ben Blonder, Rob Robichaud, and colleagues that we were involved in. But the most recent results that I just want to share briefly with you is that the adaptive radiation itself appears to be more constrained than we thought. In terms, what we found from these latest results was that even though there's been a huge amount of change, the ecological transitions have been in the direction of dry to wetter habitats. So dry to wet and wet to bog, with very little to no evidence of movement from bog to wet or wet to dry. So it appears that these plants have had difficulty evolving dry adaptations, which could be due not necessarily to the inability to evolve those traits once lost, but to competition with dry adapted members of the group that are already present. So there could be a priority effect there. But in any case, this does indicate that even this magnificent adaptive radiation does have some limitations ecologically. And that may explain why this tree, the largest in the Silver Sword Alliance, endemic to outer slopes of Haleakala, Deboutia reticulata, has ecophysiological properties intermediate between other rainforest trees in the Silver Sword Alliance and these xerophytes that grow very nearby in dry habitats on Haleakala. And Rob Robichaud actually suggested many years ago that this might reflect a recent descent of this rainforest tree from dry adapted relatives. And that's exactly what our data suggest has happened. And finally, just a really cool ending note here with regard to the Hawaiian evolution is that on the summit bog of Waialeale, arguably the wettest place on earth on Kauai, in a brief moment where the clouds parted there are 4 different sympatric or co-occurring taxa of the Silver Sword Alliance, including this amazing cushion plant, Deboutia waialeale, Ken Wood that I mentioned earlier from the National Tropical Botanical Garden, and I visited there by helicopter and were able to collect extensively on a dry day, one of the few days that when it's not raining, and found hybrids in all combinations among these four taxa that didn't seem to reflect any ecological gradients. But what we found now from this recent data is that all four of these taxa evolved bog adaptation independently of one another. So it could well be that their adaptations to this bog habitat are quite different and are not completely recombinable. Otherwise, we might expect that all these would basically devolve into a single species by forming a hybrid swarm. So our most recent conclusions are that there's been more out of Kauai dispersal than we thought, and even some back dispersal, limited to older islands, and that habitat shifts have been predominantly, if not entirely, unidirectional from dry to wetter situations, with bogs being sort of the ultimate dead end, which has been found in some other groups as well, this bogs being evolutionary dead ends. And as I mentioned, we're trying to get data from a lot of nuclear genes now across the entire tribe Madiinae, including the lily sunflowers and et cetera, to look at additional evolutionary, ecological, and biogeographic questions. And I want to end on a conservation success note. If I were to talk more about the Hawaiian flora in general, it would be exceedingly depressing because extinction in the Hawaiian biota in general is Hawaii has some of the highest extinction rates in the world. And to some extent, there's an ecological meltdown occurring due to invasive species, and particularly invasive mammals, which destroy the native plants, many of which have lost their defenses to mammalian herbivory, very much like island tameness in animals. But there are conservation successes within the Silver Sword Alliance. When I first started studying this group in the 1980s, these two taxa, the Madiinae silver sword here on the left and the MAHALO silver sword on the right, endemic to the Big Island of Hawaii, were on the very brink of extinction. But I'm talking about 24 plants in both of these species, at least within the wild populations. Today, there are tens of thousands of both of these that occur in native habitats throughout the range. They're protected from grazing by exclosures, and they're reintegrating with the native biota there. And it's really the Silver Sword Foundation, one of the real kind of the crown jewel of Hawaiian plant conservation, headed up by Silver Sword researcher Rob Robichaud, has done this work with a lot of effort, really hard work, and a lot of genetic information to make sure this is done right. So with that, I just want to acknowledge all Hawaiian researchers and all Hawaiian Silver Sword researchers for their collaboration and contributions, and all these individuals for their help in my particular work in various ways, in addition to co-authors I mentioned earlier. So sorry to go long. I just had a lot to talk about. I thought you might be interested in, and happy to consider any questions anyone has.
That was fantastic. We were on the edge of our seat here, really. I mean, what a wonderful combination of like natural forces that you brought into your story there. You know,
Oh, thanks a lot.
bird migration, you know, as well as of course adaptive radiation of the botany. Really great story, man.
Oh, thanks. I think a lot of these same themes apply to California tarweeds within California as well. And those are other stories that, you know, we could talk about another time.
Thank you. Yeah, I know it's getting really late where you're at in Michigan there, so we won't want to keep you too long. There is Orr does have one a nice question in the Q&A here, and he's asking, how do you assign genes to one genome duplicate or another in a polyploid?
Yeah, so that's a really good question. And so the main, so to do that using modern methods, there's a lot of new techniques that have come about with when we're looking at large numbers of genes, but in the way it was done in these earlier studies was kind of by brute force, really, by cloning out all of the copies of a particular gene, which picked up copies that were similar enough that both genome copies were possible to clone. And then basically just in treating those gene copies as individual taxa or species in a phylogenetic analysis with all the diploid species. And what happens when you do that is that the gene copies basically fall out where they should with regard to relationships. And that's what allowed us to see that those genomes were not each other's closest relatives, but those two Hawaiian genomes were each more closely related to different lineages of California tarweeds. But the details of, I mean, it was really a brute force operation with regard to the cloning. The cloning was done for within the Madsbox floral homeotic gene family, and similar genes were cloned, and the two different versions of those genes and the different genomes were not so distant, so divergent evolutionarily that they couldn't be recognized as variants of that gene. Sorry, but there are more sophisticated methods now with say target enriched data where you're sequencing large numbers of nuclear genes where you can phase the alleles from different genomes. And there's a software that's very helpful for doing this, like Hybridization, that can be used for that purpose. That's exactly what we're doing now with the silver sword target enriched data for a large number of nuclear genes.
Neat. And if Yvonne is asking if you might discuss the different adaptive benefits the different chloroplasts offer in the species you mentioned.
Oh, yeah. So they don't necessarily even offer different benefits. Yeah, the fact that the chloroplast genomes may be so readily transferable between species might reflect a lack of real phenotypic effects on the phenotypes of the individuals that bear them. But there are examples that are known and are documented where different chloroplast genomes do impart different physiological properties in the organisms that possess them, the trees, the plants that possess them. So it is conceivable that there could be selection for particular chloroplast genomes in particular environmental settings, and that could play into the picture of what we were finding, or it could just be a matter of the nuclear genome being more resistant to gene flow from other species due to nuclear genes having more of an effect on the overall phenotype of the plants involved, or the overall, functional ecology of the plants. So it could go either way, but that's a really great question. We don't have the answer to that for these plants.
Well, we're going to let you go on that one, Bruce. And
there's some stuff in the chat here. I don't know if that's, is that
they're mostly their thank yous primarily.
Oh, okay. I didn't open that up.
Someone and I'll save those and share them with you. Many thank yous from you're from admirers. And,
my pleasure. And I admire all of you for everything you're doing in California botany and California plant conservation and beyond. Yeah, it's an amazing community we have in California.
Yeah, we're lucky. We're so lucky. And I mean the diversity of plant species here, it's mind-boggling.
Yeah, it's just a never end. I mean, just new species constantly being described. It keeps me busy as the convening editor of the Jepson Flora Project, just trying to add, newly described species to the Jepson e-flora.
Yeah, well, we appreciate your work, and we really hope to see you again and hopefully soon. Have a great time in Michigan.
Thanks a lot. Yeah, thanks. I really appreciate it. Great to have this opportunity, and take good care.
Take care. Bye-bye, everybody.
Bye.
Good night.
Good night.